Tonight, we're going to be hearing from a six-pack of rabid scientists, and I'm including myself in that number. All of us share a common love for science and for finding out how the world around us works. But our backgrounds are very diverse, from the most philosophical to the most empirical, from the most mathematical to the most biological. We'll be here tonight to talk about intelligent design, molecular machines, and to answer all but the stupid questions that you have tonight. And as Craig mentioned, my name is Jed Macosko, and it is my privilege to be able to introduce to you the other 5 members of this six-pack. 2 of us have been asked to give in-depth talks tonight, and I'll be introducing those 2 first. The other 4 of us will be around after the break to answer questions. So first, I would like to introduce to you Dr. Fritz Schaefer. Dr. Fritz Schaefer, like me, attended MIT, then went on to Stanford first. I'm thinking of myself here, where he got his Ph.D. in a mere 2.5 years. He was immediately hired by Berkeley, which is very unusual for somebody finishing their Ph.D. And he spent time there from 1967 to 1987, roughly. In 1987, he went to the University of Georgia, where he is now the Perdue Professor of Chemistry and the director of the Center for Computational Quantum Chemistry. He's the author of over 1,000 peer-reviewed articles. Jaws drop. Oh man, I don't know how you do that. And was the 6th most cited chemist in a pool of over 600,000. That's better than the people on The Apprentice, just so you know. So that's very good. My favorite fact about Dr. Schaefer is that he was a 5-time nominee for the Nobel Prize, which is kind of the scientific equivalent of being nominated for the presidential candidate. So you can kind of imagine what kind of a guy this is, and he's going to be here tonight sharing about his love for science and his desire to see more people with a theistic mind, with a theistic worldview in the sciences. So let's welcome up Dr. Fritz Schaefer.
Thank you, Jed. Good to be here. I've known Jed for a number of years, and I've known his father for much longer. And his father and I are almost born on the same day, same year, close. You're a young guy, 4 or 5 days younger than I am. I think so. So thanks to Jed for all the hard work. And for Lynn Pooley. Is Lynn here? Is she willing to identify herself? Maybe she's so exhausted she's gone home. Anyway, thanks to Jed and to Lynn for arranging this wonderful meeting. My hat goes off to Phil Johnson. I wanted to check this with Phil for sure, but I am the person in this auditorium, I think, who has known Phil longer than anyone else, including his wife, Kathy. I met Phil in 1977 when we were both faculty members at Berkeley, shortly after Phil had become a Christian, and our friendship has endured and flourished over the years, and I'm very, very proud to consider him one of my closest friends. Here's my title for tonight, and as Jed has indicated, my goal is at least in part to appeal to the young people here with the thought of a certain sort of missionary work, namely being a missionary to a secular university as a science faculty member. So we're going to talk about that a little bit. This is also the title of a book that I've just written this year, and I think Phil will appreciate my saying that This book began in an irregular way. I was teaching freshman chemistry at Berkeley for the first time. I think it was the spring of 1983. I hadn't done this before, even though I'd been a faculty member at Berkeley for 14 years. And the freshman class is 680 students. Actually, that's how many we put in the auditorium on the first day. We know that 100 and some will disappear, so they have to sit in the aisles and things like that for the first class. And I hadn't never taught a class of 700 students before, so I was a little concerned beforehand. But I had the lecture very carefully organized, indeed choreographed. Now, the wonderful thing, I'm telling Jed something he already knows, but nobody else knows this here, I think. Or maybe Phil does. The Physical Sciences Lecture Hall at the University of California at Berkeley has a stage divided in 3 parts. You remember this, Jed? And it's awesome because 1/3 is exposed to the audience, and the other 2/3 are beyond the curtain, the veil, as it were. And the thing that's so great about this is you can be preparing your demonstrations for, well, at least 1 hour in advance, and usually 2 hours in advance, and then at the magic moment, the stage, somebody presses a button, the stage twirls around, and there you are in front of some vast number of students all ready to go, to knock their socks off. And the thing that was particularly nice about my first semester teaching freshman chemistry was that there was nobody in the auditorium the very next hour. So I could be talking to the students, and my assistant, Lonnie Martin, a bachelor's. Lonnie is still there, too. Boy, that guy just keeps on going. Lonnie could be working behind the stage and getting something really neat set up, and then at just the right time, I'd holler to Lonnie, he'd press the button, and he'd twirl around. And when you do that the first time, you know, students have never seen it. It's a pretty awesome experience. You know, you see the professor sort of disappearing into the black hole behind and then you see somebody else coming up. So I had myself perfectly ready to speak for 45 minutes on the concept of the mole, for those of you who are into that kind of thing. Very advanced concept in freshman chemistry. The idea was that when I'm done with my 45 minutes, I'm going to call to Lonnie. He's going to press the button. He's going to come around, and he's going to display something totally awesome. He's going to have a dozen different samples of common chemical substances, 10 moles of each. Obviously, water, mercury wasn't so expensive back then, maybe some lead, ethyl alcohol, of course, the reagent of most interest to the Berkeley students. And so Lonnie was going to come around, eye-popping experience for these kids. I was going to explain, I was going to get back on his part of the stage and ask the incredible question, what do all these compounds have in common? And they all look quite different, and the answer, of course, is they all have exactly the same number of molecules, and illumination was going to come over the students, and the rest of the semester was going to be a huge success. Well, I finished in 40 minutes instead of 45, my prepared remarks, and I really didn't have anything else to say. So I called to Lonnie in the hopes that he would have the moles ready and we could spend some time describing the moles, and nothing happened. Nothing happened. Lonnie's a really nice guy. I was surprised when nothing happened. Jed knows Lonnie. He's a good guy. So I hollered out a little bit louder, Lonnie, bring on the moles. Students still don't know what's going to happen. So I haven't spoiled, I mean, they know there's somebody named Lonnie somewhere around there that's going to do something, but that's about it. No response. Well, then I started walking around to the side and said, Lonnie, what's going on back there? And he said, Prof, you said 45 minutes. The moles are going to be ready in 5 minutes, just like you ordered. And I said, well, what am I supposed to do? And he said, well, shuck and jive, Prof. So this was not choreographed. I told the students, well, let me share with you something that happened to me in church yesterday morning. 680 students were very quiet. I said, I was in church and I was in Bible study, and they became even more quiet. That really did. And I'm looking at these kids, and they're thinking, oh, we've heard there are all these nuts at Berkeley, and here he is. And I said, I had shared with the Bible class that I was going to be speaking before 680 students, never had a class of more than 200, pretty concerned about it, and thinking I would get some sympathy, possibly even prayer. No. The leader of the Bible study, and I'm telling all this to the freshmen, I'm trying to do what Lonnie told me to do. This is just all coming out spontaneously. And I said that the leader of the Bible study told a story about how his freshman chemistry instructor beat his wife, kicked the dog, and every other bad thing you could do. So not exactly a jump start. And then he proceeded to ask the rhetorical question, which was very new at the time. This is 1983. The rhetorical question being, what is the difference between a dead dog lying in the middle of the road and a dead chemistry professor lying in the middle of the road? And these students, they just started to roar. They didn't know what the answer was, but just the concept of a dead dog lying in the middle of the road was a tremendous thing. And that took a couple minutes, which is good because I was running out of things to say. And when they settled down a little bit, I explained that the Bible teacher had said the difference between a dead dog lying in the middle of the road and a dead chemistry professor lying in the middle of the road is that there are skid marks in front of the dead dog. And by the time they stopped laughing, Lonnie was around with the moles. They were there. But I was looking out at these students saying, I'm not sure I'm going to like this class so much. I'm trying to imagine in my own mind what they're thinking. And it became clear to me, they're thinking, if this guy were run down on the last week of classes, it probably doesn't have anybody else that can make up a final exam. They'd have to pass us all, and we could just skip, the hardest part of the semester. Well, we did have the moles. It went down well, and class was over, and about 100 students came down to the front, and 50 of them wanted to know the answer to the canonical question, which hole do we punch in this registration card in order to get in freshman chemistry? And I was able to answer that question fairly well. And then those 50 went off. There's 50 left. And I'm wondering to myself, what are these 50 people doing? And one young man comes right up in my face. Berkeley students can be pretty obnoxious. And they're very polite at the University of Georgia. Let me just say that. But as I was telling somebody here today, I've taught more than 10,000 freshman chemistry students at the University of Georgia and not one football player. This kid comes right up in my face and he says, Prof, there's something I need to tell you. And I said, well, all right. He says, the most inspiring person in my life, the person who has most inspired me all my life was my high school chemistry teacher last year. And I said, we don't get a lot of stories like that. And I said, you have really made my day. This is just a great thing. And he said, well, that was the good news. Are you ready for the bad news? And I said, well, that news was so good, I can probably take the bad news. He said, the bad news is that this guy that I idolized told me it was impossible to be a scientist and a Christian. And I wasn't quite sure where you were going with your comments here, but it seems like you think otherwise. And I said I did. And he was not very happy, but he wandered off. Now there are 25 left. Okay? A young lady comes up, and thankfully doesn't get as close as that guy was. I have been married for 37 and a half years, and there's several people here who know my wife, so no racy stories are going to be told here tonight. This young lady comes up and says, Prof, we could use you. Well, if you're at Berkeley and somebody says something like that to you, you need to hang on to your wallet or be very cautious. And I said, okay. And, she seemed friendly. I said, what do you have in mind? Who's the we, and what sort of use are you planning to put to me? And she said, well, I'm one of the leaders of a group called InterVarsity Christian Fellowship, and we want you to come and explain to our group the answer to that young man's question. Is it possible to be a scientist and a Christian? And I said, what do I have to do? I said, well, you just show up, get ready to talk for about 40 minutes. I'll take care of the auditorium. I'll get the crowd somehow, and you do it. And that's how this began. This book began some 21 years ago. And I actually never really thought the book was going to make it until last May 23rd. I was told that I needed to report to the hospital in about 30 minutes for quintuple bypass surgery. And before they took me under, I had a little chat with my son because the book was about 80% done. And I said, Ted, my son had just graduated from Vanderbilt University, very able young man, said, Ted, if I come out on the other side of the dark river, be sure to get this book published. He looked at me with a very pained look. And I don't know if he was really worried I was going to be on the other side of the dark river, or he just didn't like the idea of writing the last 20% of the book. But in the goodness of God, I came through it. And I got out of the hospital in about 6 days. And hopefully, now Phil has had an experience like this, so he knows what I'm talking about. Basically, nobody expected me to do anything for months. Classes were over. You know, it was summertime. Nobody was expecting anything. And I decided that, you know, I really wasn't confident how many more days I had to go on this planet. So in 2 weeks, I finished the book. And then I'd been thinking about if I ever did finish this book, what would I do with it? And I thought, well, I'd do the standard stuff that everybody does. I'd ask Phil Johnson, my friend Chuck Colson, where I should publish it and all that. Well, I'm no longer sure I'm going to be around when the proofs roll in on this book. So I took it over to my friend George Smith at the University of Georgia Printing, and he printed the book in 1 month. And so we have a book that wouldn't have existed otherwise, put out in rather short order. Now, we are always looking for Ph.D. students. I see people on that side and on that side that are candidates. Chemistry majors, I can tell there are several of them out there. Yeah, I've had 73 successful Ph.D. students, not a one from Biola. So I hope this will break the curse. And you can see they've built a wonderful building for us there. It's a great place to do scientific research, and I hope that some from Biola will come and join us. Now, the one and only purpose of my talk tonight is to address the question that young man raised 21 years ago. Is it possible to be a scientist and a Christian? And of course, other people are more aggressive. They just come out and say things like, well, we all know that science has disproved God. Richard Dawkins, certainly one who would fall in that category. I do want to try to put this discussion in a little bit broader context before I start, and pointing out that science has had its challenges, struggles, fights with other intellectual disciplines as well. Perhaps some of you are familiar with this book called Literature by Gallagher and Lundin, and they talk about trouble between science and literature. They say because in recent history, literature has often found itself in opposition to science. Let me say it's a problem, and they are not vague about this. They give an example. They give as example Keats, famous English Romantic poet, and he didn't like science. He didn't like science. He thought that a Newtonian worldview is what we had. This was still the age of classical mechanics. Would be a world in which myth and poetic vision had vanished, and would make the universe a cold and uninviting place. And in his poem Les Mia, a famous poem, he talks about this. And here, let me give you just a little bit of this poem, that the word he uses for science is philosophy. So whenever he uses the word philosophy, he means science. Maybe I'll destroy the meter and just say science. That way nobody will miss it. Keats, do not all charms fly at the mere touch of cold science? There was an awful rainbow once in heaven. We knew her woof, her texture. She is given in the dull catalog of common things. Science will clip an angel's wings, conquer all mysteries by rule and line, empty the haunted air, and gnomed mind unweave a rainbow. So there has been a problem between literature and science. And let me point out, now, we don't have very many literature people at this conference, but we got a lot of philosophers. I mean, the woods are full of these guys, I'll tell you. And, you know, they are not my favorites among the people at the conference. I just have to be honest about it. You know, these guys give us scientists a really hard time. This is, all who are philosophers will understand this. I'm going to quote my former colleague, Paul Feyerabend, philosophy professor at Berkeley. We have him down at the bottom here, I think. Yeah, he died about 10. He was an unusual character. And so these philosophers are nodding. He was a strange character. We don't need to go into that. That's longer than the dead dog story. But is there a conflict between science and philosophy? Feyerabend said absolutely. And the phrasing of it is remarkable. He says, the rise of modern science coincides with the suppression of non-Western tribes by Western invaders. The tribes are not only physically suppressed, but also lose their intellectual independence, are forced to adopt the bloodthirsty religion of brotherly love, Christianity. Yeah, he doesn't mince words. He said, today this development is gradually reversed, but science still reigns supreme. He does not consider this to be a good thing. Thus, while an American can now choose the religion he likes, he is still not permitted to demand that his children learn magic rather than science at school, and yet science has no greater authority than any other form of life. Is there a conflict between science and philosophy? Paul Feyerabend certainly thought so. The young man who addressed me so aggressively that morning at Berkeley could just as well have been C.S. Lewis, my goodness, 55 years earlier. Lewis writes in the autobiography of his early life, Surprised by Joy, an interesting book, talks about why he was an atheist for a long period of time before about age 29 or 30. Lewis says, you'll understand that my atheism was inevitably based on what I believed to be the findings of the sciences. And those findings, not being a scientist, I had to take on trust, in fact, on authority. In other words, somebody told me science had disproved God, and since I never took a science course in my entire life, I believed that person and thought that I ought to be an atheist. I've got way too much material here, but I don't want to give short shrift to my atheist colleagues, so let's take a juicy one here, okay? This is, many of you will probably know that Chandrasekhar was one of the greatest physicists of the 20th century, born in India, but spent most of his life as a professor at the University of Chicago, editor of the Astrophysical Journal, much correspondence of a non-controversial type between myself and Chandrasekhar. And his biography has been written by his close friend Wally, and I recommend it to everyone. This is about 10 years old now. Chandrasekhar's been dead for about 5 years. And everybody who's read this book agrees the most interesting part of it is at the end, where there is an actual interview between the author and the subject. And it starts out this way with Chandrasekhar. He says, in fact, I consider myself an atheist, but I have a feeling of disappointment because the hope for contentment and a peaceful outlook on life as a result of pursuing a goal has remained largely unfulfilled. And having been to India 6 times and China 5 times, I'll tell you that these are the goals, the great goals of the East, contentment and a peaceful outlook. So he doesn't have them. And his biographer is pretty shocked, even though he knows Chandrasekhar well. He says, what? I don't understand. You mean single-minded pursuit of science, understanding parts of nature and comprehending nature with such enormous success still leaves you with a feeling of discontentment. Chandrasekhar is holding firm. He says, I really don't have a sense of fulfillment. All I have done seems not to be very much. And then the biographer wants to lighten this up. This is getting way too heavy for the last couple pages of the book. And he says, don't you think everybody has the same problem? But Chandrasekhar is giving no ground He says, that may be, but the fact that other people experience it doesn't change the fact that one is experiencing it. It doesn't become less personal on that account. And his last statement is something for every person who wants to be a scientist to think about, no matter what their worldview is. This is Chandrasekhar's final statement. What is true from my own personal case is I simply don't have that sense of harmony, which I had hoped for when I was young. And I have persevered in science for over 50 years. The amount of time I have devoted to other things is minuscule. If you think science is the way to find a completely fulfilling life, I advise you to read Chandrasekhar's book. So this is the question we want to ask. This is the question I was asked 21 years ago, and the main question I've been trying to answer ever since. The answer to this question is yes. And just to show you I'm not making this up, let me quote C.P. Snow, very famous extended essay he wrote, 1958 I think is the date for this, The Two Cultures. C.P. Snow, kind of an interesting guy. He was a Cambridge faculty member, not entirely successful. He was a spectroscopist, which is indeed a noble calling. But he wasn't a great spectroscopist. But he was good, or he wouldn't have been on the faculty at Cambridge. He never made professor. And when he turned about 40 years old, he decided he needed to do something new. So he started to write novels. These were not filthy Danielle Steele type novels. These are good novels. Some of these are still in print. Get a hold of his novel called The Masters. It sounds like it's science fiction or something. It's a story of intrigue. There's a murder in it about life in the college system. The masters are the bosses of the colleges at Cambridge. Anyway, he says in this, he gets near the end of his career. He's been writing novels. He's much more famous for that. He's been a moderately successful scientist. He wants to bring some unity to his life. So he writes this essay relating the world of science to the world of arts and humanities. And he writes as such. He says, statistically, I suppose slightly more scientists are in religious terms unbelievers compared with the rest of the intellectual world, but there are plenty who are religious, and that seems to be so, especially among the young. And Richard Feynman, an atheist, to his dying day, refused to go to his father's funeral because a rabbi presided, and that was offensive to him. But 9 years before he got the Nobel Prize, he made this statement: many scientists do believe in both science and God. The God of revelation, he's about what kind of a God he's talking about, in a perfectly consistent way. So it certainly is possible. One could go beyond that and cite Alan Lightman, who I think has written the definitive work on this topic, Origins, Lives and Worlds of Modern Cosmologists. And I'll just jump to he took the Dan Rather approach to this question. He bought a tape recorder, traveled all around the world, talked to famous physicists, and asked them what they thought about ultimate questions. What is life? Is there meaning in the universe? Does a God exist? Is there life after death? You know, these questions that philosophers no longer ask. Well, that ought to, I guess there aren't as many of them here as I thought. That should have gotten their blood going. Okay, and Jed is telling me, watch out for the clock. You told too many stories. Anyway, I'll just take the bottom line of this. Indeed, contrary to popular myth, scientists appear to have the same range of attitudes about religious matters as does the general public. All that time getting me introduced is your time, Jed, not mine. That is your time. Okay, yeah. All right, well, we're going to skip many things. That's sad, but we're not going to skip this. This is too good to skip. Eugene Wigner, Nobel Prize in physics in the early years, just died a couple of years ago. Labeled himself, of course, he was one of these great Jewish intellectuals that came from Budapest. And you know Johnny von Neumann as well, Michael Polanyi, Edward Teller, an amazing group of people. Once he got to the U.S., he kind of morphed into Protestantism. Used to like to talk about the remarkable effectiveness of mathematics, and stated that the miracle of the appropriateness of the language of mathematics for the formulation of the laws of physics is a wonderful gift we neither understand nor deserve. So I'm going to ask this question 3 times. I'm not going to do too much else. Why might a scientist become a Christian? And my first answer to this question would be the intelligibility of the universe. It certainly points to a sovereign creator God. This comes to us from mathematical physics. The laws of nature look just as if they have been selected as the most simple and elegant principles of intelligible change by a wise creator. Belief in the intelligibility of nature strongly suggests the existence of a cosmic mind who can construct nature in accordance with rational laws. Now, I have much more on that topic, but I'm not going to do it. I'm going to keep going. You know, the best evidence is empirical. Is it possible to be a scientist and a Christian? It must be, because virtually all of the great ones in the history of the physical sciences were Christians, and many are today. And one of the wonderful things about writing this book is I have had so many people write to me who are well-known scientists that say, hey, get me in there too. Yeah. Get me in there too. I love Jesus too. I mean, that's really heartening to see that that happen. Case Decker is here at the meeting, and he is one such. Well, we're skipping many things here. It's sad to see it go by, but Jed is giving me the very evil eye. Very evil. I'm not even looking at him now. You can see that. I'm not going to look until I get to something I like here. I'm going to ask this question 3 times. Why might a scientist become a Christian? But I have to get to the second one. Y'all are missing such good stuff here. I mean, this is really sad to see this stuff go by the wayside. Jed, you know, this is all your fault. They could be okay. Here we go. They could be just taking this all in if it weren't for you. So number 2, and this I think that okay. This is my one. You know, I'm not really a member of the ID fraternity because I don't have a research program that's looking into questions like Mike Behe and Bill Dembski are. But I'm a big cheerleader for these guys. Okay. So if there's anything I have learned from these 2 guys, it is this. Why might a scientist become a Christian? Biology. The extraordinary complexity and high information content of even the simplest living thing, the simplest self-replicating biochemical system, points to a sovereign creator God. I think that's what ID is all about. Yeah, I'm gonna try to sneak in a couple extra minutes here, but I'm really about to finish, Jed. So be patient. Be patient. I do want to ask we want to spend a little bit of time asking the why question. It is empirically demonstrable that science grew up in a Christian culture. Let's ask why. I think it's important not just to state that as a fact, but to ask why. Why did that happen? Why did sustained scientific development appear first in a Christian environment first? If Christianity is true, the universe is not an illusion, but real, the product of a God whose character is immutable. This position is at variance with pantheistic notions that place inherent distrust in sensory experience in a mercurial world. Reason number 2, if Christianity is true, being divinely created, the universe is of inherent worth and is thus worthy of study. This conclusion supplants any zeitgeist, any spirit of the times, which would view science as a mere intellectual pastime. Third, if Christianity is true, nature itself is not divine, and thus humanity may be free to probe it without fear. This was an important realization in early eras dominated by superstitions about the natural environment. Worship and ultimate reverence is reserved for the Creator, not the creation, nor humans as creatures therein. Fourth, and I've got 5 points on this, and then I'm really almost done. If Christianity is true, then mankind formed in the image of God. We've heard a good bit of what that means. Today can discover order in the universe by rational interpolation. That is, the codes of nature can be unveiled and read without such faith, science might have never developed because it might have appeared impossible in principle. Fifth and last, if Christianity is true, the form of nature is not inherent within nature. Instead, the universe reflects a divine command imposed from outside nature. Thus, the details of the world must be uncovered by observation rather than by mere rational musing, because God is free to create according to his own purposes. In this way, science was liberated from Aristotelian rationalism, whereby the creator was subjected to the dictates of reason constructed by humans. Such Gnosticism, which transformed speculation into dogma, undermined the open-endedness of science. To be sure, Christianity holds that God is a perfectly rational being who cannot act inconsistent with his character, but this principle only places partial constraints on God's creative activity, which science must be free to discover in all its diversity. Now, I am just about to finish here. Okay? Now, let me get there. I really am about to finish. And this one I've asked this question twice before. Why might a scientist become a Christian? The first answer was the intelligibility of the universe in terms of mathematical physics. The second was the astonishingly high information content in even the simplest self-replicating biochemical system. And the last one is going to be the thing that Hugh Ross talked about this morning. And I know some of you weren't here, so this is the 30-second review of what Hugh had to say. Why might a scientist become a Christian? The extraordinary fine-tuning of the universe points to a sovereign creator God. And I might even use, yes, I'm even going to use one of the quotations that Hugh used this morning. He quoted Paul Davies a little bit differently than this. Davies has written a lot on this subject. The present arrangement of matter indicates a very special choice of initial conditions. Stephen Hawking, in fact, if one considers the possible constants and laws that could have emerged, the odds against a universe that has produced life like ours are immense. And Fred Hoyle, again, we heard this this morning, a common sense interpretation of the facts suggests that a super intellect has monkeyed with physics as well as with chemistry and biology, and there are no blind forces worth talking about in nature. Thank you very much for your attention.
So Dr. Schaefer has told us about how going into science makes sense for Christians. In fact, it makes maybe even more sense when you believe in somebody who has orchestrated all of these kinds of things that we would observe, both in biology, cosmology, and all the things in between. And so it's fitting now that we hear from a man who really came into this field of molecular biology without so much emphasis on intelligent design. It wasn't until he read a book by a man named Michael Denton, Evolution of Theory in Crisis, that he found out that he had, as in his words, had been led down a primrose path. And it started him on a journey to looking into the design of the kind of molecular machines that he had been learning about and had been studying. And of course, the man I'm talking about is Michael Behe. Michael Behe graduated from Drexel University in 1974. He then went on to earn his Ph.D. from the University of Pennsylvania in 1978. He had the opportunity then to study nucleic acid structure at the NIH, and that was what he went on to build his scientific career on. He was a professor at Queen's University in New York, and then in 1986, he was asked to be a professor at Lehigh, 1985, a professor at Lehigh University, where he is currently Professor of Biochemistry. And as we all know, in 1996, his book, Darwin's Black Box, The Biochemical Challenge to Evolution, came out and made a huge impact in all of our lives. Mine especially, since this is my field, molecular machines, getting to see those chapters, chapter after chapter explaining those machines and the concept of irreducible complexity. That book has been reviewed in over 100 periodicals, including the New York Times and the premier magazine Nature. And the most interesting fact, or the fact I like most about Dr. Behe, is that he has sired 9 children, which, if you think about it, increases his Behe-ness in the world by a factor of 10 if you count his heroic wife, Celeste. So let's all welcome up Dr. Behe.
Who put that microphone up there? Well, thanks very much, Jed. It's a pleasure to be here, and especially I always enjoy traveling out to California, whenever I get the opportunity. And I was here at the conference today, and was on a break, and I was just walking around the campus, noticing all the beautiful flowers and plants, and the warm wind was blowing, and it smelled so fragrant that I decided I'd call home and taunt my wife with all the fun I was having. And I phoned home, and my daughter Rose picked up and said, "Hi, Dad." And I said, "Hi, Rose, is Mom there?" She says, "Sure." And I heard in the background, ah, and slam and crash and boom. And so my wife came to the phone, and I said, "Dear, I'm having a wonderful time here." Ha, ha, ha. And so she said I could stay here. All right, anyway. This is a picture that I got on the internet of an engine room in a ship. And I thought it would be a good one to keep in mind as I go into my talk. And I've entitled it Molecular Machines, Evidence of Design. And much like what you see in this picture, I think we can envision the cell in the same way. My talk is going to have, hopefully, 2 components. The first component, this came twice, but the first component is the question, how deeply does design extend into the cell? We've seen a lot of the machines that Jed put up for us. We've talked about design in the cosmos with Hugh Ross this morning, and design at the origin of life that Faz Rana has spoken about at the conference. And we've seen pictures of the bacterial flagellum and other things. So we know that there's a lot of design, but how deeply does it go into the cell? And that's what I hope to address in the first half of the talk. And if there's time, if there isn't, I will stop after that. But if there's time, I'm going to look back at an argument I've made about design on one particular system called the blood coagulation cascade, and show you some recent comments that some Darwinian scientists have made about it, to show you that Darwinists still do not have a grip on the problems of irreducible complexity and design that we've pointed out in these past years. Okay, so first I'm going to kind of do a Cook's tour of the design argument. And the design argument, so that I can answer the question, how deeply does design extend into the cell? It starts out like this, with Charles Darwin's statement that if it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, that his theory would break down. So we can ask ourselves what sort of an organ or what sort of a system would fit that bill. And I've argued in Darwin's Black Box that one that meets Darwin's criterion is one which is irreducibly complex. And I won't bother giving a definition, but just the example that I've used many times, probably most people here have seen it, an example from our everyday world of a mousetrap. And the point of the mousetrap example is that a mousetrap consists of a number of different components. There's the wooden platform and the holding bar and the spring with extended ends and the hammer and so on. And the mousetrap needs all of these parts to function. If you take one of them away, you don't have a mousetrap that works half as well as it used to, or a quarter as well as it used to. It's broken. It doesn't work at all. So it represents the concept of irreducible complexity. And it turns out that things like this are big challenges to Darwinian theory because because natural selection needs a system to be functioning before it can select it. And yet with irreducibly complex systems, the function really doesn't appear until the system is essentially intact. And I go on to argue that not only are things like this problems for Darwinian theory, but that they are also evidence for design. They are pointers to design. And many critics have said that design is a religious argument and not a scientific argument. But I like to show the following example to try and show you why that's not the case. Whoops. Did I? Uh-oh, something got a little out of whack. Oh, I'm sorry. No, it didn't. I got a little out of whack. Okay. And the question then is, are there any things like mousetraps in the cell which exhibit irreducible complexity? And I argued yes. And here's a drawing of the flagellum, which many of you have seen before. And the flagellum is a rotary motor. Essentially, it's an outboard motor that cells use to swim. The motor turns the propeller around and around and around, which pushes on the solution in which the bacterium finds itself and pushes the bacterium forward. There's a number of different parts that it contains, a hook region, a drive shaft, the motor itself, the stator region, bushing proteins, and in the absence of any of those components, the apparatus does not work. So the question is the conclusion of design a scientific one or a religious conclusion? And I like to use this little Far Side cartoon to show why I think it's a scientific one. And in this cartoon, we've got a troop of jungle explorers, and the lead explorer has been strung up and skewered. And this guy turns to this guy and says, "That's why I never walk in front." Words to live by. Let me tell you. Now, everybody here looks at this cartoon, and you immediately realize that this was designed. This was not an accident. In fact, the humor of the cartoon depends on you recognizing the design. But how do you know that? Is it a religious conclusion? No, of course it isn't a religious conclusion. You know it's designed because you see a number of different parts interacting with each other to perform a function that none of the parts by itself could perform. Essentially, you see what I mean by the phrase irreducible complexity. Okay, so if you think there is design in the cell, as I do, and you want to move beyond that, one question you can ask is how deeply does design extend into the cell? Is the flagellum designed, and that's pretty much it, or the flagellum and the centrosome, as Jonathan Wells talked about this afternoon, and that's it? Or is there more? Well, I contend that the cell is pretty much like this engine room, and the flagellum might be this component over here. So we have to ask ourselves, what else in the cell is designed, and how would we go about determining that? And I think one good way to do it, or one question to ask, is this. First of all, let's ask ourselves, how does this get built? How do these components get put in the right order, in the right position? Well, in the case of a complex system in our regular macroscopic world, an intelligent agent brings the components over and fits them together, screws them together, bolts them together, and so on. But in a cell, there is no active agent doing such a thing. So we can ask the question, how do the components of something like the flagellum get together? Who puts them together? And there are a number of different protein parts, a number of different components that make this thing up. How do they manage to get together? Well, you have to realize that something like this, and even the movie that Jed Macosko just showed you, really doesn't do full justice to the complexity of the proteins that it pictures. They are pictured as little kind of ovals or circles or squares or something like that. But here's an X-ray crystallographic representation of two macromolecules which are binding each other. We can think of this as two proteins binding to each other, but actually one is a protein and one's something called a tRNA. But for our purposes, we'll say this is two proteins. And you'll notice that they are their shape is very convoluted, and that one is complementary to the other. And more than that, but why do they stick together anyway? You know, the back of my hand might be complementary in shape to the surface of the top of my other hand, but you know, they don't really necessarily stick together. Why would these two things stick together? Well, they stick together because they have complementary surfaces, not only complementary in shape, but complementary in chemical properties as well. So that one region that has a negative charge in one molecule will, when it binds, be opposite a region that has a positive charge on the other molecule. Two regions which are oily will oppose each other when the two bind, and other complementary parts will stick together. So not only do the surfaces have to be complementary in shape, they've also got to be complementary in physical properties. So we can ask ourselves, well, one thing we can point out is that notice when two molecules get together, there's more than one interaction which is necessary to hold them in a stable complex. There are maybe half a dozen interactions here. So in order for two molecules to bind each other, you need multiple interactions. And remember, an irreducibly complex system needs multiple parts to interact in order to fulfill the function. So we can consider not just molecular machines, not just complicated things like the flagellum, but even the binding of two proteins to each other, the preliminary step before any function can take place, simply the binding of two surfaces to each other as an irreducibly complex system. Well, okay, let's ask ourselves, how many proteins have to do this? We see big molecular machines like the flagellum, but is that common in the cell? Well, in recent years, investigators have discussed how common molecular machines are in the cell. And in particular, some work has been done in yeast, looking at essentially all the proteins in the cell and asking the question, do the proteins generally act as individual molecules or as complexes that have to bind each other before they work? And the conclusion that the researchers have been reaching is that most cellular processes are carried out by multi-protein complexes. And they go on to point out that essentially half of the proteins in the cell, close to half of the proteins in the cell, work as complexes of 6 or more proteins. And the majority, or about half of them, work as either individual molecules or complexes containing up to 5 proteins. So we can ask ourselves, should we be surprised? What does it take to form a complex? If we consider the primordial state to be individual proteins, how likely is it to be able to form a new molecular complex? Just simply asking how likely is it to bind several proteins together, let alone worrying about the function after they bind. And this slide just shows a little cartoon from that paper showing one complex that has been discovered. And this is something called the polyadenylation apparatus. And the structure of it is not really well known, but all of these proteins are known to interact with each other in the function of the polyadenylation complex. So let's ask the question, how likely is it, suppose you have two proteins and it would be good for them to get together, bind together to form a new complex, and if that happened, then it would give the cell an edge in its struggle to survive. How likely is that to happen? Well, results from immunology are pertinent to this question, and I'm going to go through a couple papers in rapid succession here. Essentially, 100 million different protein surfaces have to be searched through in order to get a protein that binds a given protein with a fair amount of stickiness, with a binding constant on the order of micromolar, for those who know what such a thing is. The bottom line is that essentially you have to go through 100 million different protein surfaces before you find one that forms a reasonably tight binding surface with a given protein. And I'm going to skip through, but there are a number of papers in the literature which come up with essentially the exact same numbers. About 1 in 10 to the 7th, 1 in 10 to the 8th proteins have to be searched through before one gets a dissociation constant in the micromolar range. Now we can ask the question, well, okay, suppose you've got your two proteins. How long would you have to wait around before you would accidentally come across two surfaces on the proteins which would complement each other in sufficient detail that you get a reasonably tight binding complex? Now, I've been doing some work along with a man named David Snoke, who's a professor of physics at the University of Pittsburgh. And I won't go through this in detail. Suffice it to say, if one takes as a model a given protein and assumes that the gene for the protein duplicated, and you assume that one needs several changes in the surface of the protein in order to develop a new functional property, such as a binding site, then perhaps this is a route in which one might be able to develop a new binding site. But you have to worry that if you've got a duplicated gene, you might get unfavorable mutations in the sequence of the gene. And if that happens, this is supposed to represent the fact that the unfavorable mutation would essentially kill a duplicated gene, so that it would be no longer available for developing a new complementary surface for your protein. And my table is not here, but this is just a little equation. It looks complicated. It's really not. Jonathan Wells said this afternoon that when a biologist sees an equation, he should hum it. This, you'd have to have multiple parts to hum. You'd need a soprano and a bass and so on. We can dispense with this. Let's just point out that the only considerations in estimating how long it would take to develop this protein binding site are the number of organisms, which is N, that are available to develop a new protein binding site, the mutation rate, the number of sites you have to change, and so on. Now let's go to the bottom line. This is kind of messed up a little bit. But this essentially is a plot of the time, which is over here, the time in generations versus the number of sites that you have to change versus the population size of the particular species. And take my word for it that a conservative estimate, if you have to change, say, 6 or so amino acid residues in one protein to make it able to bind a second protein with a reasonable dissociation constant, you'd have to have on the order of 10 to the 20th organisms in order to develop a binding site within 100 million generations. So the point is that it takes a long time. But more than that, suppose you had to form a new complex that had not 2 proteins, but 3. And remember again, in the cell, the great majority of proteins are in complexes of 3 or more. If that was the case, then you would have to change not 6, but 12 or so amino acids. 6 on the surface of one protein, 6 on another protein. And if that were the case, and it develops by the model that I have outlined very, very briefly here, it would take on the order of a lot more organisms to develop in 100 million years. You would need more organisms than have ever lived in the history of the world. So the point is, and here are some tentative conclusions, that simply forming a new protein-protein interaction would be very difficult to do and would be expected to be extremely rare in the history of life. And we're not even talking about their function. We're just talking about locating them next to each other. For a mousetrap such as I pictured, an intelligent agent might bring one component and put it next to another and screw it on. For that to happen by a blind process would require an exceedingly long time and an exceedingly large number of organisms. And the second point is that if you need 2 of these things, if you need to develop 2 surfaces, a complex of 3 proteins, and if the intermediates have no selective value, you would not expect that ever to occur in the history of life. So the point is that design extends, if this is correct, and this is recent work, but if this is correct, design extends very deeply into the cell. It is not simply the poster child molecular machines that are often associated with design talks. It's essentially virtually everything in the cell. Okay, how much time do I have, Jed? 15 minutes. 15? Oh, wow. Okay, cool. All right. Well, let me tell you about a lecture I once gave then. Well, never. Huh? I can't hear. Oh, includes questions. Oh, 5 minutes then. 5 minutes. Okay, skip the lecture. Okay, let's briefly talk about recent comments on something called the blood coagulation cascade. Here's the table of contents of a recent issue of Bioessays, which published just a couple of months ago, and it was a special issue dedicated to, yes, molecular machines. And in the table of contents, you can see, a special issue on molecular machines, the spliceosome, the most complex macromolecular machine in the cell, and maybe a dozen articles on various molecular machines. Included in that was something called the blood coagulation system, the blood clotting system. When you go in the bathroom in the morning and shave and nick yourself, and the blood comes out and bleeds for a little bit, slows down, and stops. Nobody thinks much about that, but science in the past 40 years or so has shown that the blood coagulation system is an extremely complex molecular system. And this is what the authors of that article say. And here's a little kind of a diagram of the blood clotting cascade, where this is a component called fibrinogen, which is the precursor of the clot material itself. And when it's activated, it turns into something called fibrin, which associates to form like a fisherman's net, which slows down and stops red blood cells from leaving, from bleeding out. but the fibrin has to be in the inactive form most of the time, in the fibrinogen form, because otherwise all your blood would clot and you'd kind of freeze, and that would be sad. So it actually has to be activated by something called thrombin, but thrombin can't be active all the time, or it would cleave fibrinogen, turning it into fibrin, and you'd have the same problem. So it's in an inactive form, and it has to be activated by something else, which is in an inactive form, and that has to be activated by something else, and so on and so forth. And I had written about this blood coagulation system in Darwin's black box, and said it was an irreducibly complex molecular machine and required design. And that's what I said, but a man named Russell Doolittle disagreed with me. And he wrote about it in something called the Boston Review, which is published by the Massachusetts Institute of Technology in 1997. And that wasn't good for me, because Russell Doolittle is a prominent scientist. He's a professor of biochemistry at the University of California, San Diego, a member of the prestigious National Academy of Sciences, who has worked on the blood clotting system for 40 years. And so that's not the kind of person you want on the other side. And he said this idea of irreducible complexity is wrong. And not only is it wrong, there is experimental evidence to show that it's wrong. He wrote the following. He said, recently the gene for plasminogen, and there was a typo here, plasminogen was knocked out of mice, and predictably those mice had thrombotic complications because fibrin clots could not be cleared away. Now, plasminogen is like a chemical scissors that removes blood clots once their job has been accomplished. Not long after that, the same workers knocked out the gene for fibrinogen in another line of mice. Again, predictably, these mice were ailing, although in this case, hemorrhage was the problem. Again, fibrinogen is the precursor of fibrin, the clot material itself. And what do you think happened when these two lines of mice were crossed? For all practical purposes, the mice lacking both genes were normal. Contrary to claims about irreducible complexity, the entire ensemble of proteins is not needed. Music and harmony can arise from a smaller orchestra. So his point was this, that if you knock out one protein from the blood clotting cascade, the mice have trouble. If you knock out another protein in a different line of mice, they have a different sort of trouble. But if you combine those two lines of mice into a strain that is missing both proteins, they're okay. So the argument is perhaps the blood clotting cascade could develop slowly in steps of 2 instead of steps of dozens or so, or something like that. But we don't really have to worry about the rest of the argument, though, or how he would finish that argument, because it turns out that he was wrong. He misread the paper that he cited. The mice are in big trouble. This is the paper that he was referring to. Loss of fibrinogen rescues mice from the pleiotropic effects of plasminogen deficiency. And Russell Doolittle saw the phrase rescues mice, and he thought that the mice were normal. Well, it turns out they're not. And in the abstract of the paper, the authors wrote, mice deficient in plasminogen and fibrinogen are phenotypically indistinguishable from fibrinogen deficient mice. Now, translated into English, that means that mice missing both components have all the problems that mice missing only plasminogen have, or fibrinogen have. They can't clot. They hemorrhage. Females die during pregnancy. They are not happy campers. They are not promising evolutionary intermediates. And here's a little table of the symptoms of the mice. If they lack plasminogen, they have this suite of symptoms. If they lack fibrinogen, they have this suite of symptoms. If they lack both, they are rescued from the symptoms of plasminogen deficiency, but only to suffer the symptoms of fibrinogen deficiency. And if you think about it for just a second, you see what the problem is, or see why this should be so. Plasminogen's job is to get rid of blood clots once their job is done. And if you can't do that, the blood clots hang around, they clog up your circulatory system, and you get problems. Without fibrinogen, though, you can't form clots in the first place, and you hemorrhage, and you have got problems associated with a failure to form blood clots. If you lack both, it doesn't matter if you're missing plasminogen, because you can't form clots in the first place to gunk up your circulatory system. So you are rescued, but you've still got major big problems. Now, the scientists who did this work that Russell Doolittle cited have gone on to knock out the components of other components of the blood clotting cascade, including prothrombin and tissue factor, and in each case, the mice are in big trouble, which is exactly the result you would expect if, in fact, the blood clotting cascade were irreducibly complex, as I had argued. So what do we conclude from this? The conclusion is not simply that Russell Doolittle misread a paper, which anybody can do. You know, scientists are not known for the clarity of their prose, and it takes a while to figure out what's going on oftentimes in a paper. No, the point is this. Russell Doolittle knows more about the blood clotting cascade and its potential evolution than anybody else on Earth. And yet he does not know how Darwinian processes could have produced that system. If he did, he would have simply cited the work in his essay instead of pointing to a paper that talks about hemorrhaging mice. And if Russell Doolittle does not know how Darwinian evolution could have produced this system, then nobody knows. Nobody knows. And I think that's an important thing to keep in mind, considering Professor Schaefer quotation from C.S. Lewis, in which C.S. Lewis said he took it on scientific authority what science was able to explain. You have to keep in mind, especially in this controversial area of evolution and Darwinism and intelligent design, that oftentimes the claims of what science can and cannot explain are grossly misleading. I have a lot more to say, including a recent paper, which was actually more recent than the one I just talked about. This was a paper that appeared in something called the Journal of Thrombosis and Hemostasis just about 6 months ago with this intriguing title. And I'd be happy to tell you about it, but I think I'm out of time. And so I'll just leave with the statement that, in fact, if you read this paper, you find there still is no explanation for the blood clotting cascade. And with that, I'd like to thank you very much for your attention.
Thank you, Dr. Behe. We're going to have a little break, but if there are just a few questions that you've asked your peers, if it's okay, I'll take 2 questions and then we're going to have a break. If you have to leave, leave quietly. If there's any questions to clear up what Dr. Behe said, go ahead.
Can you hear me? With respect to the clotting mechanisms in other organisms, there's hemoglobin found in some annelids and a variety of other organisms that don't have the same cascade, but they have some parts of that cascade system. At least I'm assuming they do. Have you looked at that as a part of the evolutionary thought that might be used in objection to your irreducible complex system?
Back to me? Thanks. Yeah, that's a good question, but it turns out that the clotting system is exclusive to vertebrates. All vertebrates have a very similar clotting system. Invertebrates don't have it. And I'm sorry?
Even with the fact that they have hemoglobin.
They have hemoglobin, but hemoglobin is not a part of the clotting cascade.
And their blood's not the same.
That's
okay.
right.
Okay,
and what you should remember is that Russell Doolittle knows all about every vertebrate whose blood clotting system has been investigated. And he, even with such knowledge and the similarity between all vertebrates, is still unable to account for how the clotting cascade could have been put together.
Okay, the last question here before the break.
couple short questions. One, you mentioned binding between proteins, and it seems to me that that's not a qualitative trait, it's a quantitative trait, and therefore it could happen gradually. And it seems that there's a lot of experiments with random peptide libraries and things where they get binding experimentally under short short amounts of time with mutation selection. And there's a lot of recent examples of enzyme switching substrates, so that's one. The second one is blood clotting. It was mentioned briefly, but it seems that lampreys and critters like that have only part of the blood clotting cascade. So how do you explain that if all the parts are supposed to be there? And then with the bacterial flagellum, how do you explain the fact that almost all of the components have identified or probable homologies with systems that aren't in the flagellum with other systems?
Okay, good questions. I'll see if I can remember them all. I'll start with the last one first. An important thing to remember is that sequence homology, and for those who don't know what it is, that's the similarity of two proteins in their amino acid sequence to each other. Proteins are polymers of amino acids, and sometimes half of the residues are the same in one protein as with another protein. Sequence homology does not tell you how Darwinian processes could have put together complex molecular systems such as this. Again, just as, don't take my word for it, Russell Doolittle knows all about the similarity in amino acid sequence between proteins in the blood clotting cascade. And yet, even with that knowledge, he's completely unable to come up with a scheme for how such a thing might have originated.
I disagree on that point because he's published some papers that seem to be a pretty good scenario to me. And I've also got one by Davidson et al. In the Journal of Thrombosis and Hemostasis, 2003. I think it was about 6 months after the Aird paper. And they've got their last conclusion is, in this chapter, we review the biochemical evidence, molecular cloning, and sequence data indicating the structure of the blood coagulation network in non-mammalian vertebrates and present an evolutionary scenario to account for the possible evolution of what has been described as an irreducibly complex system that could not have arisen by gradual step-by-step Darwinian processes. And that last bit is a quote from Darwin's Black Box. So they're basically saying, here's his book, but we're refuting it.
Well, that's fine, but in my experience, the scenarios that Darwinists have come up with have been extremely sketchy, unbacked up by experiment, and essentially, I had a slide here, essentially what are called just so stories. So I'm not surprised that Darwinists can put together a story in which they declare themselves satisfied. But I think the reason I bring up Russell Doolittle's example is to show that he envisioned a scenario. He thought that if one were missing a couple of components, you could still have a reasonable blood clotting cascade. And he was utterly wrong. He was utterly...
but that was just one example where he misread a paper. I mean, he studied a lot of
Okay,
organisms.
we're going to take a break now, and the two of these guys can fight it out up front. We're going to be back here at 8:47, right on time to the schedule. So please return to your seats at 8:47 on that clock in the back. Thank you.